Hello, Dr. Williams here again with another video for you to help your revision for biology. This one is for AQA GCSE biology, and this is the biology only content, which we call triple science pathway. You should have watched the B7 combined science video before this one, as this builds on that knowledge. So this is the extra knowledge you'll need for biology. Okay, let's have a look at where we're going to get started. Okay, so the first thing you need to know about in this topic is the idea of food chains and food webs. So this builds on the combined science content, but for this particular aspect of triple, you need to know that each level of a food chain is given a trophic level. Don't get confused with tropic level. Tropisms are different to trophic. So you must say trophic because this means like an eating or feeding level, whereas the tropisms were actually all to do with B5 unit in homeostasis, and it's how a plant responds to growth towards light or against gravity, those sorts of things. So in terms of a trophic level, the first level is actually the producers. So we have a trophic level 1, which in this case is the algae, and that's our producer. The trophic level 2 is the next consumer, the primary consumer. Level 3 would be the secondary consumer, and trophic level 4 would be your tertiary consumer. We don't often get beyond 4 trophic levels. It's quite uncommon really in a food chain because we are going to get energy losses along the way. So you can see at each stage of the food chain, we should have trophic levels being passed on to consumers, but we also have decomposers at play because whenever these organisms will die or the waste that they produce, the decomposers sort of sit underneath all of these trophic levels. And decomposers are organisms, microorganisms that secrete enzymes into the surroundings in order to digest dead plant and animal matter. This makes them small and soluble, so they can diffuse into the microorganism, and the microorganism can use it for their own growth and respiration. We can look at these different feeding relationships by looking at pyramid of numbers and pyramids of biomass. And there are some examples of pyramids of numbers and pyramids of biomass. The pyramid of numbers shows you how many of each organisms there are in each trophic level. And you can see that pyramids of number, there are 2 different examples given there, one of which has got a very large base showing like a large numbers of producers and the first trophic level. For example, that could be grass, and then all of the consumers are stacked above it. In this lower pyramid of number at the bottom here, you can see there's a very small number of producers in this particular pyramid. So this could be something like an oak tree, for example. And you'd only need one of those because it's got such large biomass. So this is where a pyramid of biomass is actually better than a pyramid of numbers, because the biomass pyramids always look a pyramid shape. So the pyramid of numbers and biomass can look the same shapes, even though they might represent different things. But a pyramid of biomass is better representation of how the energy and the biomass is transferred at each trophic level, because it measures the actual mass of living matter. Remember, we remove water out when measuring biomass, and so it measures the actual matter, the actual number of biological molecules made by an individual organism or many organisms within a species of whatever you're measuring. So the pyramid of biomass always has a proper pyramid shape, whereas a pyramid of number may not. It may be kind of a pyramid that's got a very thin base. And you can actually construct a pyramid of biomass. So here's an example from an exam question where you get the biomass given at each trophic level, and you can see it's in grams per meter squared. So they've obviously got algae, invertebrate animals, small fish and large fish. You can see those numbers are decreasing. If you're asked to construct a pyramid of biomass, you need to follow these sorts of rules to make sure that you do it correctly. So the first thing is to label your scale correctly and label the axes with the word biomass and obviously the units that are given for you to use. And once you've done the scale labelled correctly, see how you start at 0 from the middle and you work either way. So as long as you've got enough to fit 840 for the algae on there, then you'll be fine. So think about your scale carefully, because remember the biomass scale is going to go totally from the middle of the pyramid either side. Then you will need to draw some accurate bars. So you can see algae is 840, so you'd have to use the scale to draw the bar at 840. So that would be 420 this side of 0, 420 this side of 0, so that makes 840 grams per metre squared when you add them up. And then you carry on with the invertebrate animals, small fish and the large fish. And normally, if this was being marked as an exam question, you would get a couple of marks for doing that accurately. Then the last stage, and this is often the one that's missed by students, is to label the bars with the different organisms that you've drawn on them. So label them with algae, and then you've got your invertebrate animals, small fish and large fish. And if you follow that method when you're constructing a pyramid of biomass, you should be able to get all the marks for that sort of question. Okay, when we think about why we get a pyramid of biomass in that shape, it's actually because there are losses in biomass at each trophic level. If we think about how much is being transferred, only 1% of the sun's energy is transferred into biomass, into the algae. Most of the sunlight will pass through the algae, or it won't hit any chloroplasts, or it won't be used in the photosynthetic pathway. It might be reflected off it, and all sorts of different ways that the sunlight energy is going. So only 1% is actually used by the algae. And then only about 10% is transferred at each trophic level beyond that. So 10% of the algae's biomass is transferred to the krill, and 10% to the salmon, and 10% to the bear. So the biomass shape always looked like a pyramid because of these biomass losses at each trophic level. So there always needs to be enough biomass to sustain the next trophic level, which explains the pyramid shape. And the pyramid of biomass shows that this biomass is lost at each trophic level, and normally this is linked to how energy is being released, and that the fact that energy is dissipated at each level as well. And these losses are due to the fact that not all material is ingested, so it's not all taken into the body and into the cells, and not all ingested material is even absorbed into the blood. So you're not getting, ingested means eaten and digested. So for example, you won't eat the bones or shells of some animals, whereas being absorbed is you might eat it, but it might not be digested by enzymes in your digestive system, so therefore isn't absorbed into the blood. That some absorbed material is lost as waste, so anything that you do manage to get absorbed, you're actually going to use in respiration, or make urea, for example, when you are breaking down proteins and things like that. So some of the absorbed material might be lost as waste, and large amounts of the glucose that you are going to absorb is going to be used in respiration so that you can release energy to carry out all of those functions required. So you can see that there are losses at each stage. So this is why a pyramid of biomass always looks the same shape. Now, in the combined science, you obviously look at decomposition and its role, especially in the carbon cycle. In the triple science, you need to know about decomposition and its role in decay and the conditions that it needs to occur. So there are 3 abiotic factors that we need for decomposition to occur. If one of them is missing, then we don't get decomposition, or we slow down the rate of decomposition dramatically. So those 3 conditions are, we need enough water for decomposition, oxygen, because obviously that is needed for aerobic respiration, and warmth is the last way to remember it. So you can think of WOW, water, oxygen, and warmth. So we basically need warm enough temperatures for decomposition to occur. And as I said before, food especially can be preserved by removing one of these factors. That's why we put things in the freezer, because we might remove the warmth from any decomposers, so food will last longer. And decomposers work by releasing digestive enzymes into the surroundings. So by releasing them into the surroundings, the enzymes are actually digesting insoluble molecules into soluble molecules so that the bacteria can themselves absorb it. So when we get the bacteria absorbing soluble molecules they need in order to survive and grow, they will leave some excess enzymes in the surroundings and excess soluble molecules. These can be absorbed by plants, especially things such as nitrates that might be left in the soil. And these plants can absorb the nitrates as part of their mineral ions in order to form amino acids and proteins and enable their growth. So decomposers play an essential role in maintaining food chains and webs and making sure that we have enough biomass at the start of all of our pyramids of biomass. And we can measure the rate of decomposition and how quickly it might happen by, normal methods of measuring rates, essentially. That's rates of chemical reactions. So we can measure it by the change in mass of something like your food source. And if it's a rate, it's got to be over a given amount of time. So we can actually measure the rate of decay. For example, of that apple there, if we measured its mass every day and recorded that, we could measure the rate of the decay of the apple. And so this then leads to a measuring decay required practical. This is an example where you can just use lipase as an example of an enzyme that digest. You could really measure that it's really an enzymes practical. If we're looking at like decay in general, there's lots of enzymes taking place in order for that to happen, but we are just looking in this case at the enzyme lipase. So we can add lipase to milk, and lipase acts on lipids and turns them into fatty acids and glycerol. Fatty acids, given their name, are acidic. So the pH of your milk solution will actually decrease. And then you can change different variables. So you could change, for example, the temperature. And here you can see plots plotted for different temperatures and the time for a colour change for an indicator like phenolphthalein or other types of indicator. When that colour change occurs, you can see how long that takes. And that will obviously tell you when a certain threshold of fatty acids are being released. So this is a way of measuring the rate of decay. There are other methods that we can use to measure rate of decay. It's unlikely you'll get a start to finish, let's write up this practical, but you might be asked, and this draws actually in on knowledge from B2 about enzymes and also knowledge from rates of reaction. So that's C6 in your chemistry as well. So just apply those ideas. But we can use decay on a day-to-day basis. So gardeners make use of organic waste material to make compost, and you might have a compost bin yourselves. And you need to make sure that compost bin is well aerated, so it's got lots of oxygen, so that those microorganisms that are carrying out the decay have enough oxygen for respiration, that they are kept moist and warm. So remember the conditions of WOW, and making sure that all those conditions are met inside that material. And if that isn't met, then those microorganisms will not decay in ideal conditions. The compost might start to smell. You might get some more toxic chemicals being produced and anaerobic conditions in there, which isn't what you want. We can also use decay in the ideas of fermentation. So again, this applies back to, I suppose, paper 1, where you were learning about anaerobic respiration and fermentation in yeast. So modern biotechnology, you can grow microorganisms on a large scale, and essentially what you're doing is controlling their decomposition rates. And when we decay in this type, it's called fermentation. We can do this in aerobic or anaerobic ways, and you can do it large or small scale. So some examples of fermentation: you can make beer at home, and that's using fermentation by yeast. You can also, like fermented foods are quite popular these days, so you can carry out fermentation in jars. And you can also have these really large scale fermenters at like brewery factories and places. And a fermenter, when you use a large scale fermenter, is a piece of equipment where decomposers can decay organic plant matter. And they can do this with or without oxygen. And you must regulate the temperature carefully, because if you think about decay, it is going to involve respiration, aerobic respiration or anaerobic. They are both exothermic reactions. So you're going to get heat produced. So what you need to do is regulate the temperature. So you might have to cool it. So in this example here, you've got cooling water surrounding the fermenter. If temperatures get too high, it's really important that we make sure that the temperature is maintained, because the proteins could end up denaturing. And those proteins, if they're enzymes, then you're going to get respiration basically stopping. And so the whole process will stop and the decomposers will die. So you've got to regulate the temperature. Other things in a fermenter that you need to regulate are the gases. So you can't let gases build up. If gases are being produced by aerobic respiration, you're going to need oxygen going in, but you also need a way for the carbon dioxide to leave the fermenter. Any gases released need to be released into the atmosphere because pressure will build and then your lid might explode off the fermenter. So you can see you've got, with this fermenter, you've got an exhaust outlet so the gases can leave. And you've also got a way for air to get in. And if you want your fermenter to work properly, you need to continuously stir it. So you have these stirring paddles, and that allows the nutrients to be mixed up so the microorganisms don't need to rely on diffusion over large diffusion pathways. We've got a shorter diffusion path because you're mixing them directly next to those nutrients. So you're getting the microorganisms with all the nutrients that they need to carry this out effectively. We can adapt fermenters to grow in different types of microorganisms which will carry out decay to produce different products. So one of the types of fermenter you might come across is fermenters used for biofuel, where we get microorganisms grown in anaerobic conditions, and this can produce methane gas. So you basically get the anaerobic decomposition happening. You use manure, for example, from cows, and you might have the decomposers there decomposing in anaerobic conditions to release methane gas, and that gas can be collected and used as a fuel. And this is how biogas generators work. And the biogas that produced can be also called a biofuel. So this is a key method of, really, it's an effective way of recycling waste materials and getting some useful biogas from it. Another type of fermenter, which again makes biofuel, is using yeast again. And yeast can be used in anaerobic fermentation to produce bioethanol. So specifically bioethanol because it's anaerobic. And if you remember the anaerobic respiration equation for yeast, you will get glucose converted into carbon dioxide and ethanol. So this is where the yeast can be used, and we can collect the ethanol, and it's actually called bioethanol, and that can be used as biofuel. Another type of fermenter you might come across is a mycoprotein fermenter, and a mycoprotein actually doesn't use bacteria in there, but it uses a fungus called fusarium, and this fungus is grown on a glucose syrup in aerobic conditions, so you need to make sure that oxygen goes in there. And the mycoprotein produced by this fungus can be extracted, and this is often used in meat-free foods. An example of corn is given there. So corn is just mycoprotein from fungus from fusarium. And all of these methods can be used to kind of help us to secure food. So thinking about using the fermenter effectively, can think about how we can use biofuels effectively, but also things like using mycoprotein instead of meat products can actually help with our food security. So food security means having enough food to feed our populations. And the following things can affect food security. So birth rates increasing, so that's going to obviously more people to feed. It's more difficult to provide enough food. People have changing diets. So some countries food is scarce, and in other countries food is transported a long way. New pests and pathogens can affect farming. There's an example there with the banana. There's a fungus affecting the banana species at the minute. And because there's not much variety, this is really affecting the production of bananas. Environmental changes that affect food production. So if rains don't fall, if you have droughts, or if you have flooding, this can really have a big impact on the food production for those countries. And because food is shipped so far in the world now, this can impact food in a lot of places. The cost of agricultural inputs, so the cost of fertilizers and all of those resources you might need to grow food, and as well as conflicts can have a big impact on those agricultural impacts and energy supplies. So this can limit the amount of food available for people to eat. So with the food we've got, we need to make the most of it, really. So farmers and companies are trying to increase the yield all the time of the foods that they grow. When this is animals that we are farming, then we need to make sure that we are getting the most from those animals that we can. One of the ways is to make shorter food chains, so they are much more efficient because there's fewer energy losses. So if we can get animals to eat plants directly without having to feed them extra resources, then that's obviously going to be a shorter food chain. So there is less energy lost. But you can also restrict animal movement and help to keep them warm, so they're not having to use respiration in order to regulate their body temperature. You can try and feed them more protein so that they build up more biomass as well, but obviously there's a cost there. That protein ideally should come from plants rather from other animals. And there's also a question about the ethics around this. If you're restricting animal movement, I don't think there's a problem with regulating temperatures, but really restricting animal movement, whether that's ethically appropriate and kind to those animals. If we want to increase the yield of fishing, fishing presents a certain issue because it's difficult to farm fish. So we have to regulate the numbers of fish that we're allowed to catch in order to make sure we have food security around the amount of fish that we are eating. So lots of fish stocks are currently overfished. Here you can see tuna and mackerel populations declining. And this really is being driven by industrial fishing, so large-scale fishing, illegal fishing where it shouldn't be happening, it is, unreported fishing and unregulated fishing, then that's going to obviously impact how well we can maintain our fish stocks. So some things we can do to help our fishing populations and be able to use fish as a food source is to make sure that fishing is sustainable. So we can report and use media to put pressure on industry and governments. Mesh size is quite an important one. If you increase the size of the mesh, then we are only going to catch the larger fish. The smaller fish can swim through and they can carry on to grow into adulthood and breed and reproduce their own fish. If we are catching the larger fish, they have probably already bred. So there's certain sizes of fish that you are allowed to catch. And you can have fishing quotas, so there's certain numbers of fish that you are also allowed to catch. And obviously these steps need to be put in place because, for example, the quota of Arctic cod was much greater in 2018 and now it's much, much smaller in 2025. You can actually farm fish though, and salmon fish farming is quite common. And this involves raising salmon in little hatcheries, so they are surrounded by nets, and they transfer them into these nets when they're about 12 to 24 months until they reach the market size. You can see this is a major source of industry. Not everywhere in the world is able to do fish farmings, but as the UK have got a large coastline, it's especially important in Scotland and Norway. However, there's some criticism with environmental impacts because sometimes the fish can be infested with sea lice. Now, obviously, these sea lice, they're going to feed off the salmon that are being farmed and they are going to reduce the biomass of the salmon. So thinking about the welfare of the fish as well as the actual yield that we get from fish farming is an important consideration, and how much time and effort we have to spend removing those sea lice. They also use wild fish to feed the salmon, which obviously is not a great solution to the food security issue or the fish quota issue either. But there are other methods of producing protein, and this links again into B6, where we looked at genetic modification. So genetic modification is where we can increase yield by adding certain genes into plant and animal crops in order to make them maybe more resistant to disease or drought or contain extra nutrients. So we can make sure we are getting lots of yield. So that's an example at the top of golden rice, which has got a vitamin A gene added into it. So that's one strategy. Other strategies can be used with biotechnology. So when we are looking at those fermenters and growing things on a large scale and using yeast and things like fusarium to make proteins, that's obviously another method of making sure there's enough food. And then scientists are looking in ways of using artificial meat and growing them into tissue culture, so basically growing meat into dishes. That's still in an early development phase, but you can see that that would be a good alternative because it's not cruel to animals, because there would be no killing of animals involved. Okay, that's it really for all the triple content for B7. It's really important that you understand the fermenter section of this. Those questions often come up, so I would recommend you do a few practice questions on those. But again, it's about applying your ideas, reading question carefully, and really thinking about the big picture a lot of the time in a lot of these issues. So good luck with any exams you've got coming up.